Hello everyone, I'm Paul Warren, the solo finalist from Charles County, Maryland. My experiment, the physiological effects of microgravity and increased levels of radiation on wild-type and genetically modified Caenorhabditis elegans, was inspired by a technology, entertainment, and design talk given by Cynthia Kenyon on how a mutation in the DAF gene lets C. elegans, a model organism in biology, live up to 100% longer. This mutation works by decreasing the sensitivity of cells to insulin-like signaling. My experiment will measure the impact of multi-generational C. elegans growth on the various physiological properties of this common nematode. C. elegans has a short life cycle, a compact genome, stereotypical development, easy, rapid propagation, and is both extremely small and very common. It has around 1,000 somatic cells and can be easily mutated. In addition, it is hermaphroditic and self-fertilizes. A large amount of progeny is produced per adult. Its entire genome has been mapped, and the development of every one of its somatic cells has been traced. These characteristics make C. elegans a model organism not only in microbiology experiments on Earth, but also in space experiments as well. As many as 8 previous spaceflight experiments run by real scientists from all over the world have used C. elegans. My experiment uses a type 2 fluid mixing environment. The type 2 FME has one long ampule contained in the main compartment. I've used heat to sterilize the FME via a machine called an autoclave. In the long ampule, I've placed a liquid growing medium called Caenorhabditis elegans habitation and reproduction medium. This medium provides the sustenance and living environment for the nematodes. Also in the long ampule are approximately 8,000 different C. elegans of 2 different strains. The first strain is a wild-type strain, pretty much what you would find in the wild. The only difference is that this strain has a muscle defect and an added green fluorescent protein for easy identification. This strain will serve as a control group, as I'll be able to gather data from this strain and compare it to data gathered from past experiments and determine whether or not my experiment was actually scientifically valid. The other strain is the long-living strain I mentioned earlier. This is the strain I'm really interested in. And not only will I be able to look at the effects of the environment of space on this long-living strain, past spaceflight experiments have determined that the DAF16 gene, which is again the gene that extends lifespan when mutated, is also linked to the majority of other gene mutations. So my experiment will not only be able to answer my original question of the effect of the environment of space on the lifespan of these worms, but may be able to answer questions that are currently at the forefront of space microbiology. Now, the reproductive lifespan of C. elegans under optimal lab conditions is around 180 hours. That ensures that growth will be exponential. Because of the size of the FME and oxygen restraints, only enough media to allow for 3 weeks of growth can be used. As such, the astronaut broke the ampule 3 weeks before the experiment left the ISS, mixing the starved larvae and the media and beginning growth. 7 identical experiments were run on Earth at the exact same time under near identical conditions and were opened periodically to give clues as to the actual experiment's progress. Now, I've been given an amazing opportunity through this experiment. Thanks to the help of Dr. Nathaniel Shefchak of the University of Nottingham, a professor and fellow spaceflight researcher who has helped me a lot on this experiment, I'm now the head of an international group of scientists from the University of Nottingham, the National Institutes of Health, the JAXA, the Japanese Space Agency, and Tohoku University in Japan. I worked closely with Dr. Shefchak, his chief technician, Dr. Freya Shepard, and 3 scientists from the National Institute of Health, Dr. Andy Golden, Dr. John Hanover, and Dr. Donna Love. I've learned a lot from these scientists, and it would not have been possible to come this far without their help. One of the biggest things I focused on is learning how to do what I want to do with the worms when they come back. I want to compare the relative reproductive strength of the 2 strains using a machine called the worm sorter to gather quantitative data. I want to track movement autonomously using a program called the Worm Lab. I want to analyze the genome of the worms and various protein sequences, and hopefully I'll be able to determine the effects of the environment of space on the lifespan and brood size of these worms. If all goes well, my results will be published in an actual scientific journal. Now, obviously, I've been able to do some pretty amazing things with this experiment, and I have to thank everyone who's been involved. Sometimes I wake up and still can't quite believe that I, a 16-year-old high school student, have sent an experiment, and not just any experiment, but a scientifically valid experiment, an experiment that may be at the forefront of space microbiology, aboard the world's first commercial spaceflight to the International Space Station. I can't thank all the wonderful folks at SSEP enough. Dr. Goldstein and Dr. Van Hale especially, not just for giving me the opportunity, but for all the support they gave me during the actual experiment. Everyone at Charles County Public Schools has also been amazingly supportive. My teacher facilitators, Laura North and Romulo Gabriel, have helped me a lot, and I would not have been able to go to NIH as many times and do as much with the experiment as I have without the support of James Short, the principal, the rest of the administration, and everyone at the Board of Ed. I've also been given the fantastic opportunity to put my NIH-taught worm knowledge into actual practice at a local microbiology lab at the College of Southern Maryland, an opportunity I have to thank Ms. Terry Jordan and the rest of the staff for. The scientists at NIH and everywhere else have been amazingly supportive, as I mentioned before. Again, I'd like to thank Dr. Nathaniel Shefchak for all the support he's given me. And finally, last but not least, I'd like to thank the two people who have raised me to question endlessly and helped me push myself to accomplish what I've accomplished so far. I'd like to thank my mom and my dad. Without you two, this would not have been possible, and I can't thank you enough. You two are the absolute best parents a kid could ask for, and I really appreciate everything you've done for me. Thank you.
Questions for Paul?
You stated that you had two types of these nematode species in this in the same container. Were you worried about contamination with the strains?
No.
Like crossbreeding or?
No. Again, they self-fertilize. Mm-hmm. And even if they do cross-contaminate, it would be pretty easy to actually eliminate the strains that have been mixed because, like I said, the wild type has a muscle defect, and the other one has a gene that's been deleted. So all I can do is run it through the machine that I'm using and set it to eliminate the mixtures.
So were you looking at some certain variables to test the microgravity or just the general growth and reproductive cycles?
I'm going to look at the relative reproductive strength, first off, using the machine up there called the worm sorter. What that means is when the experiment comes back, I'm going to pretty much use the machine to count all of the worms. And because of the 3 days, that it's going to be not in space, what I'm going to do is set the worms so that it doesn't count the worms that have actually just been grown in the past 3 days. Then there's the movement tracking and the genome and the protein sequences and the general lifespan and brood size as well.
All right, thanks.
Yep.
Well, I had a question. You said something in your presentation that I just wanted to go back to. You said that you are leading an international team of professional researchers.
That's correct.
Can you tell us a little bit about how you put that team together?
Well, it's like I said in my presentation, I really have to thank Dr. Nathaniel Shefchak for giving me the opportunity to do all of this. What he's done is he's helped me refine my experimental design and helped me get into contact with all of these scientists. He actually flew over from England, from the University of Nottingham, to meet with me and my teacher facilitators, and to go up to NIH and talk to the scientists there to figure out if I would actually be able to go there and work in their labs. And as a result of that meeting, I have been working at the National Institutes of Health for the past 5 or 6 months.
So what are you expecting to see in terms of the effect of microgravity on the lifespan of the worms?
Well, what I'm expecting to see is a significant change. I don't know which direction the change is going to have. They might not live for as long as they have, or they might live for even longer than they have. Because when I first started this experiment, I expected there to be a slight change in the experiment because of microgravity, and I really wanted to figure out, the muscle defect that I have in the wild type, that might be repaired in space as well. But as I was doing more research, I found out that the strain that I'm talking about has never been sent in space before, but the gene has been shown to be related to all of the genes that have been mutated as a result of spaceflight. So they send normal worms out, and they come back, and they count all the genes that have been mutated, and they can link all of those genes to my gene, the gene in the strain that I'm sending up. So I really have no idea which direction it's going to have. I'm just hoping it has a very significant change.
Thank you.
Yeah.
Thank you.
All right.